Hello. Today we'll be doing the first episode of a new series. It's basically about the recent advances that happened. So the first topic that we're going to discuss is about emerging roles of glycocalyx. Now, for the gist of it, this one particular article is what I refer to. So a major chunk of what I'm going to discuss today is going to be based on this. So you can feel free to go and read it if you want. Um, yeah, and apart from that, few other diagrams and few extra points I've added it along. So let's start. So this is an introduction from that article that I just told you, and this is pretty much enough for you to understand what basically a glycocalyx is. Okay, so the literal meaning of glycocalyx translates to what is called sweet husk. Okay, so sweet husk. So it's sweet because it contains sugars, monosaccharides. Yeah, and it's a husk. It kind of surrounds the cell like a cloak. So it's like a rice and outside the covering of it. So it's like a cloak around the cell. So next thing you need to understand is what is a glycan. So these are actually conjugates of sugar. Okay, so these different sugars in the glycocalyx are connected to each other. Okay, and they form a whole network, and this connected version is what is a glycan. Okay, so this sugar conjugate is what is a glycan. And next thing is you have to understand that these glycans can be different types. They can be glycoproteins, proteoglycans, they can be glycolipids. So based on whatever it is, yeah. So this term glycocalyx, when we refer to it, we actually use it in a broader term. Okay, it's an umbrella term that includes your free glycans, the glycoproteins, the proteoglycans, glycolipids, which are there on the cell surface. So if you want, this is a good enough definition of what a glycocalyx is. Now, once that you understood that, we'll take a diagrammatic representation of it and try to understand. So this is a cell membrane. Consider these to be integral proteins. Okay, so there are proteins which are there, and to the proteins there is a saccharide unit which is attached. Okay, now compare these two, you'll see that this is much longer. So a protein to which some 220 saccharide unit, yeah, so oligosaccharide is attached, that is what is called the protein part, is the major part. So that is a glycoprotein, because protein is the major part. Over here you can see a huge polymer. Okay, so here the major part is actually the saccharide part. So because of that, this is what we call a proteoglycan. So this is the glycan part is more compared to the protein part. So this is a proteoglycan. Next, let's just consider this to be lipid. So if that is a lipid, what is attached to it? Glycan again. So that is your glycolipid. And of course, apart from all these, we have free, yeah. So we have free glycans which are not attached to anything. So this whole thing together is what forms your whole glycocalyx. Okay, so you can see a cell, and all around the cell, this will be like a dense network. Okay, which kind of protects the cell actually. So that is your glycocalyx. So this is a diagram from that article that I was talking about. You can see that proteins are there attached to the proteins, lipids are there attached to the lipids, and we have free glycans also, which are all there together just above the membrane. Okay, for forming a whole coating around the cell. So you can see another version of glycocalyx here. So this is your endothelium, blood vessel endothelium. You can see this fine hair-like projections of this is your glycocalyx. Okay, so magnified image of that. You can see the endothelial cell here, and this is your glycocalyx structure that you see there. Now again, I want you to take a guess about what is the glycocalyx over here. Okay, so in case you thought this was a glycocalyx, no. Okay, this is actually your intestinal mucosa, which have villi, microvilli, and all that. So that is what you see over here, and this is your glycocalyx. Okay, so above the cell, outside, there's a glycocalyx layer over there. So we'll come to understanding the functions. What is glycocalyx? We already understood that. Okay, what exactly does it do? So all the primitive concepts that we have, some new things that we come to know about recently, we'll just discuss about all of that. So base thing is, protection. So this is your cell. All around the cell it comes like a coating. So basically what does it do? It prevents the entry of pathogens from outside to a degree. Yeah, even nanoparticles for that matter have been prevented from entering inside. So this ultimately leads to protection. Now when you talk about vascular, so we had just seen a diagram of your vascular endothelial and the covering over the glycocalyx. So what it does is it kind of prevents underside leukocyte adhesion, underside adhesion of platelets. So when you learn all this in different chapters in immunology, or when you talk about coagulation, all that, you learn all this basically. So this is what it does. And apart from that, it acts like a mechanosensor. So because blood is a flowing thing, okay, so this is often referred to as brushing. Okay, they appear like bristles and wind passing through, making them rustle like this. So that is how the picture is usually given. So based on that, that will lead to intracellular responses, vasodilation, actin, cytoskeletal rearrangement, and all that. Okay, so you can take a look at this diagram. This is the glycocalyx that you see outside. You can see the RBCs over here, albumin and all that. Yeah. So generally, this is most likely going to be long polymers. Okay. So things like heparin sulfate, chondroitin sulfates, proteoglycans like this, and you can see the hyaluronic acid. So these are the things which are there predominantly in the glycocalyx of endothelial cell. And over here, what you can notice is, because the endothelial layer has been disrupted, there is increased leukocyte adhesion and thrombus formation. And another interesting thing that has been noted is that once the blood flow has been interrupted, it's seen that the glycocalyx seems to shed. Okay, the exact mechanism is still not clear, but this is something we noticed. So another thing about the function is that it acts as a storage compartment for galactins and other proteins. So we'll just briefly try to understand what a galactin is. Okay, so galactin is actually a protein. Okay, this protein connects to beta galactosides. Okay, so this structure has two sides where it can connect to your beta galactoside. So either it can connect using two sides to beta galactoside, or they can use these sides to connect amongst themselves. Okay, so what it does is it kind of forms a whole complicated lattice sort of a network because of that. So they kind of bind multiple glycans, interconnect them, and the proteins to which they attach. And this kind of leads to clustering of bound glycocalyx components and formation of the so-called galactin lattice. Okay, so these substances, there are different types of galactin 1, 3, numerous types of it. Okay, so they are known to play multiple roles in our body, like morphogenesis regulation, organization of primary cilium, attenuation of apoptosis, endocytosis. So these galactin seem to play such roles, and they are intimately related to your glycocalyx. Okay, and these things, based on whatever signaling is happening, they can store these galactin in the glycocalyx, and when needed they can actually bring it back into the cell to use this. So galactin 1, just for this morphogenesis regulation, galactin 1 actually is taken all the way to the nucleus for that, and the rest of the functions are actually galactin 3's function. Okay, so this is another thing that your glycocalyx does. Next is to control cell morphology. So the component called mucin is what kind of regulates this. So you can see that this is the structure of mucin. Okay, so this is a O-glycosylated protein backbone which is attached to a membrane anchor. So there's a membrane anchor to which it's attached to the cell membrane. You can see the protein here, this black thing over here is protein, and all the blue thing, that is a O-glycosylated. These O-glycosylated protein through a membrane anchor is attached to the membrane. This thing, okay, so you can see how a cell is flat and adherent, but when the mucin expression is increasing density of mucin expression, that kind of changes the shape to lift it up. First thing is it got lifted up, and there's increased tubularization that you see over here. Okay, so all this is kind of, this entropic penalty that is happening. To reduce it, all this happens. Okay, so let's go to the next one. Next is regulation of membrane protein diffusion. So this is where they use a term called the picket fence model. Okay, so picket fence, this, in Tom and Jerry, you must have seen this. Okay, people in India don't usually see this. So this is what is called picket fence. So basically there is a wooden structure like this, attached like this, and anchored down like this. Okay, so in our membrane there are proteins like this. Okay, so these proteins, they are CD44. So CD44 acts as this protein. Okay, and it is anchored down through esrin onto your actin, the cytoskeleton structure, and on top you can see these attachments here. That is your hyaluron. So this hyaluronic acid is here, that is there outside. CD44 is there, through esrin it is attached to your cortical cytoskeleton, the actin. So there's a whole complex like this. So what this complex does is, it also helps in some sort of a protection and all that. Okay, it makes things difficult to pass through the membrane. But when you talk about things like macrophage, which needs to phagocytize, so consider there's a pathogen outside, and this thing needs to go and phagocytose it. So this is the leading edge, and this is the trailing edge. Okay, the leading edge for the pathogen to be, taken in, and for the receptor to, get to understand what it is, and for it to mobilize, you need this structure to be broken down only in this area, not over there in the trailing area. Okay, so that is what happens in the macrophage. The breakdown of this structure results in receptor mobilization. Okay, and that will be higher over there, and that results in engulfing. So this is another interesting that happens because of the glycocalyx. Glycocalyx is also seen to be involved with, cancer. What is seen is that, normally when the glycocalyx is thin, the integrin, okay, so when the glycocalyx is thin, integrin seems to jut outside. So integrin, as you know it kind of adheres to your extracellular matrix. So it connects the cell to extracellular matrix substances like the fibronectin, collagen, and all that. Okay, so that is what integrin does. Now, if about proto-oncogenic events, yeah, either it's epithelial mesenchymal transition or oncogenic RAS activation, whatever, it kind of increases the width, okay, of your glycocalyx. Okay, so once your glycocalyx becomes more thicker, then the integrin is no longer outside, it's inside, so it becomes difficult for it to interact with the extracellular matrix. So that interaction, signaling, everything is not happening properly. So this can keep growing. It can even help metastasize without any anchoring anywhere. Okay, or to tell the limit of when to stop moving, when to stop growing, and all that. Okay, so that is what happens. So this kind of increases the cancer and helps it prevail. Okay, next thing is in immunology. So in cancer cells, it's seen that there are these sialylated proteins and lipids. These sialylated proteins and lipids in these cancer cells, they bind to, in an immune cell, what is called the Siglec. So sialylated proteins and lipids bind to the Siglec in the immune cell. So this kind of activates a lot of things. I'm not sure, I don't know what level of students are watching this, so let's not go too much into detail of this. Okay, that's a whole other topic by itself. Okay, so a tyrosine based, yeah, so either inhibition motive or switch motive, whatever, it's a tyrosine based thing that happens, because of which it activates a lot of things. Okay, and ultimately what it does is it kind of reduces the activity of your natural killer cells. Okay, so you saw what happens because of this cancer cell. It has ability to inhibit your natural killer cells activity. So this is actually a therapeutic target if you think about it. Okay, so yeah, so let's just recap the functions of your glycocalyx. One is for protection, yeah, we have seen that. Vascular, mechanosensor, then of course the leukocyte adhesion is prevented, and adhesion of platelets, all these which are not desired. Okay, then storage compartment for galactin and other proteins, control cell morphology through mucin structure, and regulation of membrane protein diffusion. We have talked about the picket fence pattern, yeah, and the CD44, and the functional relevant for cancer development at all stages. So we talked about the length of the glycocalyx, okay, and then the immune system regulation and checkpoint inhibition. So that is where we talked about the silylated proteins and lipids. Okay, so hope this was helpful to understand something new that is happening. Okay, so thank you for watching. See you around.